While I think setting up his presentation, I will take the opportunity here to welcome everyone, including Dr. Niranjan, to this webinar on the subject of Salmonella diagnosis, infections, current challenges and future directions. A little bit about Dr. Niranjan. Of course, from Uttar Pradesh, he needs no introduction within the state, but for those of us who are interacting with him for the very first time, I'm very pleased that he's able to take time out from his busy schedule and join us today. He's a medical microbiologist with more than 12 years of academic, diagnostic, and research experience. He has been in the field of medical education for a very long time, imparting education in medical microbiology to students of MBBS, BDS, pharmacy, and other health sciences. He is, as he describes himself, a solution-oriented gentleman, and has been involved in clinical diagnostic services and infection control programs in various hospitals that he has been assigned with. Also trained in tropical medicine and specializes in infectious disease control. He is at present the consultant clinical microbiologist and group infection control officer at the Regency Group of Hospitals of both Lucknow and Kanpur. So he has, to his credit, more than 15 papers, research and publication papers covering a varied gamut of subjects. And we are so happy that Dr. Niranjan could take time out today from his schedule and join us for this webinar on the subject of Salmonella diagnosis. So without further ado, Dr. Saab, over to you. You can share your screen, run the presentation that you're in.
Just give me 1 minute. I am not able to open that PowerPoint, which I have shared.
Sir.
So I need to call the IT team for that.
Otherwise, I can help you out here, sir, if you are okay with it.
I think that will be better if you can share.
What I'll do is I'll share it from my side. Whenever you want me to change the slide, just say next. Is that okay?
Okay, that will be. Thank you.
No problem, sir. None at all. No problem at all. Just give me a second. I'll just download.
Sure.
Right, sir. Over to you, sir.
So good evening all. And so to start with this topic, diagnosis of Salmonella infections, current challenges and future directions, I would like to thank JMitra for giving me opportunity to interact with the people around the world, and also thank you for the kind introduction. And this Salmonella infections, basically there are two types of infections which we need to diagnose, and there are various challenges that we are facing. And current time, when there is rain continuous and Salmonella infections can be there, so we need to understand. So my talk will go from introduction to Salmonella, then its methods of diagnosis, and then what are the future, what are the challenges we are facing with the current methods of diagnosis, and what are the future directions for that. Next, please. So this is a gram-negative bacteria which causes enteric fever, which commonly people known as typhoid fever. It can also cause gastroenteritis and septicemia. There are more than 2,000 serotypes which are from the single species of bacteria, and it can be typed on the basis of serotyping and species typing. Next, please. So when we see the bacteria, they are classified into various families. So when we see this bacteria belong to family Enterobacteriaceae and to tribe Salmonella, and genus is Salmonella. which is again speciated into Salmonella enterica, Salmonella enteritis. So Salmonella enterica is further divided into their serotypes. These serotypes are typed, so that name that we know as Salmonella typhi, in this Salmonella is the name of genus and typhi is the name of serotype. So complete name, when we pronounce it, it becomes a long name. Salmonella enterica, subspecies enterica, serotype typhi. So this long name cannot be used, cannot be user friendly. So we use in the short form that Salmonella typhi. Similar is for paratyphi A, B, C, Salmonella typhimurium, Salmonella enteritis, and Salmonella choleracis. This Salmonella typhi causes typhoid fever and paratyphi ABC causes paratyphoid fever. And these collectively known as enteric fever. While those diseases caused by enteritis and typhimurium are gastroenteritis. And cholera Swiss usually causes bacterial septicemia only. Next, please. Next. So when we look on the clinical syndromes, before diagnosing any disease, we need to understand the syndromes caused by these bacteria. So number one is typhoid fever, that is enteric fever caused by Salmonella typhi, Salmonella paratyphi ABC. Salmonella, other serotypes of this Salmonella like typhimurium, enteritis, Newport, Dublin, Thompson, there are more than 2000 serotypes. They all causes gastroenteritis. Cholera Swiss causes septicemia. And there is another spectrum of these diseases to be a carrier stage. Carrier stage is found in those who carry the bacteria in their gut, but they are not affected by that bacteria. So they don't have symptoms of this infection of this bacteria, but they can transmit that bacteria to other persons. Next, please. So when we talk of source, number one source is food for Salmonella infections. Out of this, raw and undercooked meat, which includes chicken, turkey, duck, beef, veal, and pork. There are raw fruits and vegetable, which is unwashed and uncleaned. Unpasteurized milk and other dairy product of those cows or those buffaloes who are infected with Salmonella. That also includes soft cheese, ice cream, and yogurt. Raw and undercooked eggs, processed food like chicken nuggets and nut butters. So they all can transmit Salmonella into our gut. Other methods of transmission includes poor hand washing and from the pets. Poor hand washing is when we see other than the food source, poor hand washing becomes the another important cause of transmission. And animals, pet animals like dogs, cats, birds, and reptiles, they can carry the bacteria in their gut, and their droppings, when contaminate our food, they can cause the salmonellosis. Next, please. So when we talk of Salmonella poisoning or food poisoning caused by Salmonella species, so acute symptoms when patient comes to you will comes in cramps in your stomach, bloody poo, diarrhea, cold and chills, fever, headache, upset stomach, and vomiting. These symptoms usually starts within 8 to 72 hours of infections, and most symptoms subside by themselves and patient can recover within a week without any additional treatment. Until unless patient is going into dehydration, we need to treat that. Next, please. The problem comes with invasive salmonellosis. Invasive salmonellosis means the bacteria is into our blood. It is caused by Salmonella enterica, serotype typhi, and paratyphi A, B, and C, which causes enteric fever. Salmonella paratyphi can also cause septicemia and metastatic purulent infections. So metastatic purulent infections means there is infections, multiple purulent infections, which can move from one place to another place. Invasive non-typhoidal Salmonella, that includes Salmonella enteritis and Salmonella typhimurium. And highest burden of these invasive salmonellosis found in immunocompromised and malnourished individuals, especially the children which are infected with HIV. And among these, those patients who are immunocompromised and have septicemia because of and in invasive non-typhoidal salmonellosis, they can even prove fatal to those patients. Next, please. So when we see this bacteria and we do the gram staining, we found they are gram-negative bacilli, means they are rod-shaped bacteria, and they are facultatively anaerobic. They are motile, non-sporing, and non-capsulated. And when we see the motility pattern, it is because of peritrichous flagella. That is, flagella is all around the body of bacteria. There are two species of Salmonella which are non-motile, that includes Salmonella gallinarum and Salmonella colorum. And they can be distinguished from other bacteria by biochemical and antigenic structure that we will be discussing during the diagnostic part. Next, please. So when we look for lab diagnosis methods, number one is culture. That remains the gold standard for invasive Salmonella infections. And culture is isolation of bacilli in the lab and then testing for antibiotic susceptibility for that bacteria. Serological tests that include demonstration of antibodies, demonstration of circulating antigens in the patient, and other lab tests that can correlate with the Salmonella infections. Molecular methods that includes PCR, RT-PCR, and there is one multiplex PCR that is coming up, that is BioFire, which can be utilized for diagnosis of Salmonella infections. But that is seldom needed in cases of Salmonella infections. Next, please. So for isolation of bacteria, it is done by culture of specimen like blood, feces, urine, or aspirated duodenal fluid. Out of these, most commonly used samples that we receive in the lab are the blood and the feces. Urine we rarely receive, especially in those cases which are carrier of Salmonella, and we want to diagnose carriers. Aspirated duodenal fluid is also used for same purpose. Relevant specimen is required because correct specimen gives you the correct diagnosis. If a specimen is not right, then diagnosis will never be right. So when we analyze the errors in the reporting, then pre-analytical errors are the most common errors. So if a patient is in the first week of illness, then first choice will be the blood culture, which have positivity percentage of around 90%. So in 90% cases, we will get a positive culture when we collect the blood in cases of Salmonella infection. In second week of disease, blood culture is again on top, then followed by feces culture, and then Vidal test, but that will show the low titer. In third week, blood culture, feces culture, and Vidal test almost as equal, if percentage around 60 to 80%. In fourth week, it becomes Vidal test becomes the top priority, or the Salmonella IgM testing that becomes. Blood culture percentage drops to 25%. Next, please. Bacteremia means presence of bacteria in the blood of patient. That occurs early in the disease. So when we see first week, second week, and third week, it is 90%, 75%, and 60% positivity. While fourth week and later, it becomes 25% positivity. Next, please. So how blood culture is collected? Blood culture, to collect blood, we need to ask the patient that we are going to collect your blood for culture method, and that will include approximately 8 to 10 ml blood per bottle, and at least 2 bottles collected. So that 2 bottles are very minimum 2 bottles. Otherwise, it is advised to have 2 to 3 sets of blood culture, because when we do the blood culture, we need to culture at least 0.01% of the blood. So when we calculate the blood volume of the person, that is adult, for adult, it is around 5 liters. So we need to culture at least 50 ml of blood. When we have to, so for that, at least 2 sets or 3 sets when we collect, then positivity increases to greater heights. And we collect the blood aseptically. That is very, very important, because when we are collecting blood for blood culture, if it is not collected by aseptic method, then that will lead to contamination, and contaminants will overgrow and pathogens will be lost. So we will not be able to diagnose. So aseptic method, we collect the blood and send to the lab. Lab incubated at 37 degrees centigrade, for 5 to 7 days. Usually Salmonella becomes positive, blood culture becomes positive by second or third day. So it requires incubation around 24 to 36 hours and then it becomes positive. So basically जो manual methods हैं, they are glucose broth and taurocolate broth. Two broths are used, or otherwise we can use the glucose phosphate broth only. But that will need the taurocolate broth will help in enriching so that isolation of Salmonella become easier. So once we found them turbid, then we need to subculture on a solid media. So culturing is on the liquid media helps in us to get a positive response early. We cannot place 50 ml blood on a solid culture media. That is not possible. So that's why we are doing the liquid culture. So once liquid culture is done, it becomes positive, then we can transmit those colonies, transfer those bacteria into the solid culture media. So on McConkey agar, when we see, as you all know, that is the preferred media for gram-negative bacteria. The colonies will be non-lactose fermenting. They will be yellowish in color or colorless. Next, please. There are certain automated methods of blood culturing are nowadays available, and most of the labs are equipped with those in basically in metro cities. So these, that is BACTEC by the BD and BACT Alert 3D by bioMérieux, and there is one walk-away system by Siemens, and there are multiple these 3-4 systems are available in India, where it is automated blood culture system. Here what we do, we collect the blood culture in the special bottles provided by the company, and we incubate into that incubator, which are agitating incubator, which continuously moving the blood to and forth. So positivity becomes more. And second, they contain sodium polyethylene sulfonate, that is SPS, which can neutralize the bactericidal activity of blood and antibiotics if patient has taken prior. So blood culture positivity rate increases in that cases. So we need to, in another advantage in this is once it becomes positive, the machine flashes on the screen that this blood culture is positive. So you can directly take and subculture on the solid culture media. In manual method, you have to subculture after second day. You have to subculture every day to see whether it is positive or negative. Next, please. When we see the colonies on McConkey agar, as I have told you, they are colorless or yellowish in color. So you can appreciate these colonies on blood McConkey agar. Next, please. Another method which was introduced was clot culture. Clot cultures, they are alternative to blood culture, and sometimes they become more productive in yielding better results in isolation of bacteria. This 5 ml blood is withdrawn and is allowed to clot. Once it is clotted, then clot is lysed with the enzyme streptokinase and bacteria released from clot. And that once it is lysed with the streptokinase, that goes into the culture media, and then bacterial isolation becomes higher. But this method, issue with this method was that there was some chances of contamination. During clotting used by streptokine, lysis by streptokinase and then again going into the media. So there were chances of contamination more. Next, please. What are the challenges that we face with blood culture? Number one is availability of blood culture facilities. In tier 2 or tier 3 cities, there are hardly blood culture facilities are available, and so it becomes very difficult to get it done. And turnaround time is quite high. We need to at least 48 to 72 hours to give you a positive result, even when it is positive after 12 hours. Blood volume, that is again, at least 20 ml blood is collected for each blood culture. Prior antibiotic therapy, that can lead to false negative blood cultures. Bone marrow is comparatively more sensitive than blood culture, bone marrow culture, but bone marrow aspiration is again a very invasive procedure and require a specialized person to collect bone marrow. And that is also being invasive. So we are not doing that culture in most of the labs. Next, please. feces culture, which is again most commonly used culture after feces, after blood. It is valuable during antibiotic therapy because blood culture can show no growth of organisms and feces might show the growth of that bacteria. Antibiotic does not eliminate bacilli from the gut, while it does, it can eliminate from the blood. So what we do, feces, stool we collect, and stool is inoculated into selenite broth and taurochlorid broth, or directly plated into McConkey agar, DCA, XLD, or Wilson-Villiers media. So in our lab, we are preferring XLD media along with this selenite F broth. So we enrich the bacteria in selenite F broth. Once it is enriched for 4 to 6 hours, it is again subcultured onto XLD media. Next, please. So when we see the colonies on XLD, they are red color colonies with black centers, which the Salmonella, which are S2S production negative, that is hydrogen sulfide gas negative, they produce red colonies without black centers. On Wilson and Villiers bismuth sulfide media, they produce jet black colonies due to formation of S2S, while Paratyphi A produce green colonies due to absence of S2S production. So that will depend on, so that will also help in preliminary identification of species also on specialized culture media. Next, please. These are the colonies of Salmonella typhi on XLD agar. Here you can appreciate the pink colonies with the black centers. Black center has increased to almost covering the colony in isolated colonies, as you can see. Next, please. Other samples like urine, as I have told you, it is rarely used, but it can also be positive in 25% of cases. And especially there are two types of carrier in Salmonella. Number one is urinary carrier. Number two is biliary carrier. So biliary carrier, that bacteria lies in gallbladder of the patient carrier, and urinary carrier, the bacteria lies in the urinary bladder of the patient carrier. So for those patients, we can utilize urine cultures, centrifuge the urine, deposits are inoculated onto the plate, and then you can get the isolated colonies of the Salmonella typhi. Here you need a specialized media like XLD or TCBS. Otherwise, the gut flora will overgrow and you will not be able to see the pathogen. Other samples, as I've told you, bone marrow, bile, duodenal aspiration, CSF, and sputum. Sputum rarely becomes positive, and rarely we get pneumonia with because of Salmonella. That is mostly secondary of septicemia. But bone marrow, bile, CSF, they can come positive. But again, these are the invasive specimen, which needs a specialized person to collect them and send to lab. On average, blood and bone marrow cultures have a sensitivity around 50 to 80%, as I have already told you, which directly correlate with the number of viable bacteria in blood. In blood, there are less than 1 colony forming unit per ml, while in bone marrow, it is around 10 colony forming unit per ml. So that's why sensitivity of bone marrow is higher in cases of Salmonella infections. Next, please. A little about the biochemical reaction. If we are putting up on manual method, then how will we identify the bacteria? So we need to put up at least 5-6 sugars. IMBOK test, as you all know, indole, methyl red, BP, MR, and citrate. So this minimum is required to identify the species at least. Up to the genus level, we can identify. And by analyzing glucose, mannitol, and lactose, sucrose, sugar combination, we can identify their species. So Salmonella typhi will produce acid with glucose. With mannitol, they will again utilize it, will produce acid. They will not be utilizing lactose. So all the Salmonella are non-lactose fermenters. They will be sucrose negative and they will be indole negative. Salmonella typhi and Paratyphi A, they will be citrate negative. So they will not utilize citrate as the sole source of carbon, while Paratyphi B and C will be able to utilize citrate. So if it is becoming acid and gas production, so it can be Salmonella Paratyphi A, B, or C in case of glucose and mannitol. But when we see the citrate results, so if it is citrate positive, so it can be Paratyphi A is out. It can be Paratyphi B or Paratyphi C. then paratyphi B and C is very difficult to differentiate. So then we correlate with clinical symptoms. What are the, when we are doing it manually? When we are doing it automated system, then we can always identify them on the automated system. And second is by the serotyping. There are antisera available for that bacteria. So that antisera can be utilized to identify the exact serotype. Next, please. There are certain slide agglutination tests are also available, where culture isolate is emulsified in 2 drops of saline on a microscopic slide. And so we use one emulsion as a control so that we can compare whether there is agglutination or not. Control does not show any agglutination. It is carried out by polyvalent O and polyvalent H first. So it is O and H positive, then that increase that genus Salmonella is there. So this is serotyping method that I have already explained you. So once it is positive for O and H, polyvalent O and polyvalent H, then we can do with O9, O11, O3, O1, and H3, H1, H2, H3. So accordingly, these combinations are there, and then we utilize that table to identify that particular species or serotype. Next, please. One test which is most commonly talked, most commonly written by the clinicians, but it is the most debated test also. That is Vidal test. Mr. Vidal, who was a professor in pathology and internal medicine at University of Paris, and he developed the procedure for diagnosing typhoid fever, and it was to identify antibodies in the blood of infected individual. So that causes the binding together of the antibodies, and that will, we can see it into clumps. So that is known as Vidal reaction. Next, please. So this is basically a tube agglutination test. As you understand, agglutination means clumping of the bacteria or antibodies or antigen. So when there's clumping, that is agglutination. So when we talk about tube agglutination, so this agglutination is happening in a tube. There are 2 types of tube, Dryas tube and Felix tube, but most of the lab are using only one type of tube, that is round bottom tube. And it is used for diagnosis of typhoid and paratyphoid, because you will have that one TH, one AH, one BH. So paratyphi A, B, and typhi can be diagnosed. TO is common to all the 3 species. Testing for H agglutinin is done in Dryas tube, that becomes more sensitive, but still you can use the round bottom tube. But in conical bottom tube, the sensitivity becomes higher. In O agglutinin is tested in Felix tube, which is a round bottom tube. And then they are incubated at 37 degrees centigrade overnight, preferably in a water bath. Next, please. In first week, you will not get it positive. So it has become very imperative to train your consultants, your clinicians, that don't send Vidal in first week. Either you send in second and fourth week so that we can see the rise in titer and diagnosis can be made, or you can see after second week, at least one sample. But Vidal, serum agglutinin rise abruptly during second and third week. And 2, as I have told you, there are 2 serum specimens are preferred, 7 to 10 days with 7 to 10 days different. O and H antigen are used for Salmonella typhi, and only H antigen of Salmonella paratyphi A and B. Because somatic O antigen is common to all these 3 bacteria. Next, please. Serum dilution. We prepare the serum dilution of the patient, starting from 1 in 20 to 40, 1 in 80, 1 in 160, 1 in. So serial doubling dilutions are made of for O, for H, TH, for AH, for BH. So there are 4 rows of tubes which we arrange up to 560 or more. Then what we will see the H agglutinin first, then O agglutinin. In unapparent infection, it can be false positive, or it can be in cases of positive in cases of immunization, a patient being immunized for typhoid, or patient was previously infected with Salmonella. So that is means patient was previously immunized or previously infected, they can produce antibodies against that bacteria. So those antibodies will come positive in Vidal test. So that's why Vidal test become debatable. The patient might not be having infection, but still it can be positive. Next, please. So it is one problem is time consuming because it requires at least 24 hours. Sample comes and it is put into test, then overnight incubation, then next day it is read and report is sent. So that cannot help you to start an antibiotic regime. But still, it is the most common test which is written by the physician in India. Nowadays, they have shifted to testing for IgM Salmonella typhi, that is by RCT test. So next, please. That is detection of Salmonella IgM by ICT or typhoid antigen in blood and urine by utilizing counter immunoelectrophoresis and enzyme ELISA. These two tests are not commercialized much and they are not available also. So very rarely these tests are used, especially for research purposes only. While ICT is nowadays most common test written by the clinician for diagnosis of Salmonella. Next, please. Most common rapid diagnostic test is Tubex and TyphiDot that we use. TyphiDot, Tubex detects anti-O9 IgM antibodies in Salmonella typhi and anti-O2 antibodies in Salmonella paratyphi. So Tubex can detect the typhoid and paratyphoid simultaneously, but TyphiDot detects IgM and IgG antibodies against 50 kilodalton outer membrane protein of Salmonella typhi. Advantage is we get results early. Disadvantage is we can only diagnose Salmonella typhi, not paratyphi. A and B will be missed out. Next, please. IgM detection is most suitable marker for diagnosing acute infections, but once it is positive, it remains positive for up to 3 to 6 months. So we have to have that history also with us. Sometimes interpretation of test results is very important by the clinician or by the lab person when they are releasing the results. They must take history whether the patient was positive for typhoid in previous 3 to 6 months. Many a times patient get over treated for this reason. Their IgM typhoid is coming positive and they are given antibiotic again and again without going for blood culture or any other culture method to actually see whether it is causing infections or not. So this people who are IgM detection is most suitable marker for diagnosing acute infections among those people who have not previously been infected with Salmonella typhi and paratyphi and have not been vaccinated with TY221A live attenuated vaccine, because this can interfere with the results. IgG detection suggests reinfection in convalescent patients, infection in vaccinated people or asymptomatic carriers. These tests, they show 80 to 90% specificity and 70 to 80% sensitivity. And they have possibility of more extensive use as a point of care testing for taking the patient on treatment. Next, please. Circulating antigen demonstration, it is rapid, sensitive and specific test. It is positive during first week of disease and antigens are detected by co-agglutination test. Still, I don't think that any of the commercial kit is available with this principle. But I found it very interesting because this COV1 strain of Staph aureus, which contains a protein A, it is stabilized and then coated with S-typhi antibodies. And when it is mixed with patient serum on slide, so it gives results within minutes. So within 2 minutes, you will have result as it is positive for Salmonella typhi. It is equivalent to this rapid diagnostic test, but detecting antigen become more specific rather than detecting antibodies, because typhoid antibodies like IgM survive for 3 to 6 months and IgG survives up to 3 years. So 3 to 5 years, it keep on testing, it will keep on coming positive. So that's why this test is has more potential to detect the acute infections. Next, please. Typhoid carriers, as I have told you, 1 in 30 of the survivors who has got the Salmonella infection become carriers. And this carrier becoming tendency is, as the study suggests, is more common in females as compared to males. In carriers, the bacteria remains hidden inside the cells and the gallbladder, causing new infections as they are shed from the apparently healthy host. So these persons, when they handle food handlers become a problem. So that is a next, please. There's a story of typhoid Mary. So these patients who keep on secreting that in the stool, and if their hand hygiene is not proper, they can transmit that bacteria to other patients. So this typhoid Mary was in America, who has changed multiple jobs. And everywhere she used to go, people used to fall sick and they used to have Salmonella typhi infections. And then by the time they were diagnosed and by the time she was pinpointed for that purpose, she used to change her job and city. So that keep on doing, and one day she was caught, and she was labeled as typhoid Mary. So that's a very interesting story of community-acquired infections, and that this chronic period, we need to identify those carriers. For that identification of, and there are a lot of stress on diagnosing infections in the patients in acute infections, but carrier diagnosis is not done. Nobody care about that also. But this become important for the people who are directly handling food, like in kitchen people. Like in our hospital, we test every person who is recruited for kitchen. We test their stool for carriers. We test their urine for carrier stage, and then every 6 monthly they are tested for Salmonella carriers. So it become this is also important for those street vendors who are dispensing food in the restaurants. So this should be mandated by the government that all the kitchen staff should be tested for Salmonella carriage and other parasites. Next, please. So in carriers, Vidal test is of no value. Sewer swab technique is used for tracing of carriers. It can be used in plant cities like in America or UK or Europe. In India, there are sewage is not proper, then the sewer swab technique cannot be utilized in our place. So in this, they used to do sewage filtration through a millipore membrane. That membrane is put into the culture media. Then we need to isolate. So if it is isolated, then separate sewers on that area were tested. In किस house से आ रहा है, उस house के लोगों को test करते थे. So that's how they can pinpoint the carrier in that area. In India, we are in very infantile stage in that. Next, please. So this typhoid Mary, real name was Mary Mellon. She worked as a cook in New York City in early 1900s. She was responsible for infecting at least 78 people. Out of those, 5 were died. And these highly infectious carriers, they pose a great risk to public health. But they could do in those years. In India, we are still not able to do. Next is nucleic acid detection. It involves PCR that amplifies Salmonella serovirus-specific DNA for diagnosis. So PCR can be conventional, real-time PCR, or it can be a nested PCR, or it can be a multiplex PCR. They are advantageous because they can detect specific DNA extracted from live or dead bacteria. Dead bacteria in blood can result from antibiotic treatment, which is also common in some endemic areas. So these dead bacteria we can detect. So effect of antibiotic can be nullified. Disadvantage is the machines are costly, trained persons are needed, and everywhere it is not available. Next, please. Most common use targets that include flagellin, fleece, VI polysaccharide. VI polysaccharide also used to diagnose carriers, 16S ribosomal RNA, heat shock proteins, cytotoxins, and other conserved genes. Sensitivity depends on the sample processing and the training of the person and person who is putting up the test. It ranges from 40 to 100%, but specificity is 100%. Next, please. Future, which are under development or under research, they include protein biomarkers, nucleic acid biomarkers, and metabolite biomarkers. So we'll discuss briefly about each. Protein biomarkers, next please. So in this protein biomarker is basically possible because we can isolate whole protein of Salmonella, which is expressed in E. coli, which was probed with enteric fever patient samples to screen for the presence of specific immunodominant bacterial antigen signatures. Once it is identified, then immunoaffinity proteomics based technology, that is IPT, which was used where their columns are there, which are packed with enteric fever patient antibodies. And they are probed with bacterial antigen to discover bacterial antigen highly immunogenic in enteric fever. So if patient sample is put into that column, that will match and that will come as a band. So proteomics is the upcoming structure, like MALDI-TOF has come up for identification of bacteria that identifies bacteria on the basis of their protein structure. So that's how it goes, but can be diagnosed from directly from the blood of the patient. Serum or plasma is needed, but this is under the development only. Next, please. Nucleic acid biomarkers, they include 5 signature genes, that is STAT1, SLAMF8, PSME2, VARS, and ALDH-IA1. They can identify enteric fever with 88% specificity and 97% sensitivity. There is miniature NMR, which detect bacterial messenger RNA using magneto-DNA probes, and that has been proposed. They are so specific that they can detect up to 1 CFU per ml in Salmonella typhi and Salmonella paratyphi. So we can put these tests directly onto the blood of patient. So because concentration is around 1 CFU per ml in the blood, so it will be able to detect that. Next, please. Metabolite biomarkers, it is comparative analysis of metabolites in the patient, between the patient plasma samples who are infected with Salmonella typhi and paratyphi and control samples, and they compared via gas chromatography and mass spectrometry, and that has identified 695 distinct metabolite peaks. Next, please. When we analyze them combined, they produce 6 peaks, which can reflect ethanolamine, gluconic acid, monosaccharide, phenylalanine, piperolic acid, and saccharides. They can effectively distinguish typhoid fever patients from paratyphoid fever patients and healthy individuals. And tight correlation of these metabolites, like iron and tryptophan to typhoid fever, also demonstrated in Salmonella typhi, that can induce a rapid decline in plasma iron levels and retention of iron inside tissue macrophages through the upregulation of hepcidin. Lot of molecules which are under development. So these all future that we are seeing. Next, please. Acute infections also generate a specific interferon signature, which can alter tryptophan catabolic pathway, which leads to pathogenesis of typhoid fever. So this pathogenesis and identification, that is under development. Next, please. Now, so we have talked about diagnosis of invasive Salmonella infections like enteric fever, but when there is gastroenteritis only and there is no enteric fever, then we need, it's a zoonotic disease. Source of infection, again, contaminated animal products, poultry, meat, milk, cream, eggs. It can also occur as a cross infection from one patient to another patient. It can manifest with diarrhea, vomiting, abdominal pain, fever. Sometimes it might lead to septicemia when it is untreated and left for. Commonest species is Salmonella typhimurium in India. Other common species include Enteritidis, Newport, September, and Anatom. Next, please. Again, it is isolation by culturing stool. Rarely, patient needs antibiotic, and more frequent in developing and developed nations. Next. Salmonella septicemia It is caused by Salmonella cholera suis. It causes deep abscess in the body. Deep abscess means below the muscularis mucosa. Endocarditis, inflammation of endocardium of heart, and diagnosis is by the isolation from blood and pus. To diagnose cholera suis, I would like to emphasize here, we need at least 3 sets of blood culture. Otherwise, it might come as false negative. Chloramphenicol is the highly effective. Once it is diagnosed and chloramphenicol is started, patient respond very early. Next, please. So coming on to a preventive aspect, if we have done the diagnosis and treatment, but still we need to prevent. How to prevent? We should not eat raw or undercooked food. Cross-contamination of food should be avoided. Do not prepare food or pour water if you are infected with bacteria. Wash hands, kitchen surfaces and utensils with soap and water. Wash hands after contact with animals or animal feces. Avoid direct or indirect contact with the reptiles and infants. Next, please. So basically, that I would like to emphasize is the hand hygiene is again a most common, most efficient, and least costly method of infection control. So simple hand hygiene and washing can reduce the several cases of typhoid as well as other bacterial infections or other parasitic infections in our country. Thank you very much.
Thank you very much, Dr. Nap. That was, typhoid is one of the oldest known diseases around town. It's been around since the ancient Greeks from 430 BC. And even today, we have not found all the solutions to typhoid. I think one observation that I did have when I met a certain doctor in Bombay last year during after the second wave, and I was talking to him about incidence of various diseases. He said, Mr. Mahajan, the typhoid has reduced substantially because of people have started using sanitizers and cleaning hands.
Yeah.
It's a surprising development. Which
I would also like to share one incidence. Like in our hospital, we used to isolate around 300 to 400 Salmonella typhi in a year. But last year and last 2 years, when the COVID era comes and patients started washing hands with the sanitizers, we could isolate around 30 isolates only.
That's a 90% reduction, doctor.
That's a 90% reduction in Salmonella infection. This year, we are again getting those.
Yes, because people have lost they've lost the fear of COVID. There is no mask, there is no fear. But thank you so much, sir, for bringing, so many things to the fore again. It's, yes, I'm sure a lot of our people who are viewing it today have, because they come from the lab background or they are medical practitioners, they would know about typhoid. It's, it's something that is taught in every school. But getting a refresher course, looking at what's new around the corner. Sir, I had a question, especially with respect to nucleic acid testing, PCR, that after COVID, there has been a preponderance, an excessive availability of PCR machines in the country today, something that was not there in the pre-COVID era. So do you see any major changes happening in various forms of testing, whether it's dengue, whether it's malaria, dengue, typhoid, chikungunya? I have seen certain people moving towards PCR. Do you see any big change like this happening going forward?
See, the specificity and time both has attracted clinicians towards the going for molecular diagnosis for most of the cases. Now, availability of machines is there, but still they have a closed system machine mostly in most of the labs. Open system machines are not available, so they can utilize other kits also. So closed system are there. They have to depend on the company when they are making the cartridge for that machine, for that particular test. However, those companies have taken it as a challenge. Now they are providing cartridges for diagnosis of other infections other than COVID or TB. These two were most commonly available. Now we have for HIV, SCV, HBsAg, dengue, chikungunya, salmonella. Now, like BioFire has come up with the multiplex PCR system that can diagnose even a syndromic approach. So we do not need to put up multiple tests for identifying. Sometimes like viral infections, fungal infections, we are not able to diagnose by conventional methods of culture or by any diagnostic method, because we need to put up multiple tests. We need to have that DD in front of us. We need to put up multiple tests. but this biofire has picked up very well in most of the places, because it can give you like, it become more important in cases of CSF and pneumonia. So CSF, usually viral culture is not available in most of the places. Hardly we have 3-4 viral labs in India which can culture the virus and identify. Still the time taken is very high. So now this biofire gives you opportunity to isolate, identify that viral infections, and accordingly patient can be put on the treatment.
Right, sir. There was one question that did come in from the audience. I'll just share it with you.
Yes.
There was I don't know if it's a lady or a gentleman because the names are open. But the question was, what's the positivity of blood culture manually for Salmonella in enteric fever? So,
Salmonella
yeah,
manual culture positivity is almost similar to automatic cultures, slightly less than that, but that require multiple subcultures. Like after one day, then in the next day, and then every next day, you keep on doing subcultures. So that can lose out sometimes when you forget to subculture, and then you come to know it is positive. And second, sensitivity of manual blood culture is always lower than the automatic cultures because of the continuously agitating system that keep on agitating the blood, and blood cells keep on breaking. If it is intracellular, that can come out and can grow. But in manual, it is not there. That facility is not there because their bottles kept still on the incubator. So positivity of it is around 20-30% lesser than the automatic cultures.
right. Thank you so much, doctor. We are still we still think of ourselves in the COVID era. So for now, we will love to appreciate you here with a little, how do you call it? A certificate of appreciation, sir. Thank you so much for your time and effort and for bringing typhoid back into our mind space. Thank you, sir.
Thank you. Thank you.
Mr. Govind or Mr. Subhash will be meeting you shortly in next week with the formal hard copy of the certificate, sir.
Thank you.
Thank you for your time, and thank you everyone for attending today's webinar. We look forward to seeing you again in 2 weeks when we have the next webinar on wheat-related disorders and skin treatment. Thank you, everybody. Have a good day.
Thank you.